All right, let's dive right in. Welcome to This Explainer. Today, we're looking at a deeply fascinating but honestly profoundly unsettling topic. We're going to examine how the human pursuit of perfection, whether that's breeding for speed, for scientific consistency, pure aesthetics, or just straight up profit, has systematically destroyed the genetic integrity of the very animals we admire. We're going to trace the hidden and often ignored costs of human breeding and look at exactly what happens when we try to design living creatures to our own exact specifications. So to really understand how we got here, we first need to talk about something called the inbreeding coefficient. Simply put, it's a metric of how closely related an animal's parents are. Pretty straightforward, right? Well, what's wild is how this played out in the real world with a famous thoroughbred horse named Northern Dancer. This horse won the Kentucky Derby back in 1964 and then retired to stud. Breeders absolutely loved his offspring, so they kept using him. Then his sons, then his grandsons. And this intense drive to engineer the ultimate champion set off a super dangerous chain reaction that fundamentally altered the genetic makeup of the entire species. The staggering result of that chain reaction? 90%. Today, literally 90% of every racehorse on Earth descends from that single animal, Northern Dancer. And because of this massive genetic bottleneck, the average inbreeding coefficient across the entire breed now sits at 12 to 13%. The direct physical consequence of that? Thoroughbreds fracture their legs at totally unprecedented rates. The whole industry focused entirely on speed and size and pretty much ignored bone density. They've been trying to replicate legendary horses like Secretariat by doubling down on these narrow bloodlines, completely ignoring the basic biological reality that hybrid vigor just cannot be inherited twice. Okay, let's shift gears over to the scientific realm and see how this plays out in a completely different context. Way back in 1921, a scientist named Clarence Cook Little wanted an animal so genetically uniform that it would respond identically to tests every single time. So he bred mice sibling to sibling for over 100 generations. The result is the C57BL/6 lab mouse. It's essentially a population of living clones. Today, these exact mice are used in 25 to 30% of all biomedical research on the planet. But here's the huge catch. They have incredibly high rates of cataracts, progressive hearing loss, and bone fractures. This deliberately stripped-down, genetically flawed animal actually forms the foundational model for a massive portion of what we think we know about human biology.
Section 2, the purebred pet crisis.
We've seen what happens in sports and science, but there's a deeply systemic issue happening right in our own living rooms, with the pets we love. Take the English bulldog, for example. Bred for a flatter face and a heavier skull, to the point where an insane 80% require a C-section just to be born. Then you have the pug. Their skulls are so compressed they suffer from brachycephalic obstructive airway syndrome, which basically means they struggle to breathe just existing. Look at the Cavalier King Charles Spaniel. They were bred for this cute rounded domed skull, but now it's literally too small for their own brains, resulting in up to 95% of the breeds suffering from brain malformations and spinal cavities. And then there's the Dalmatian, whose very specific coat patterns was selected right alongside a genetic defect that completely blocks the poor animal from processing uric acid. The very aesthetic traits breeders purposefully selected for have become the leading causes of chronic suffering in these dogs. And when we look at cancer death rates, the data gets even more specific and honestly a lot darker. There's a stark 23-point difference between American-bred golden retrievers, who faced a massive 61.4% mortality rate from cancer, and European-bred dogs, who sit at 38.8%. So why the huge gap? Well, this massive discrepancy traces directly back to different founding populations. In America, a highly narrow gene pool of just a handful of champion sires spread their genetics across hundreds and hundreds of litters. Basically, somebody's favorite dog became everybody's favorite dog, and that specific genetic bottleneck is exactly what's killing them today. Now, let's contrast the original vision of a breed with its modern reality. On one hand, we have the working line German Shepherd from 1899, created by Max von Stephanetz. Notice the level straight back. It was built entirely for function and sustained movement. But then, look at the modern show line. Breeders actively selected for this dramatically sloped silhouette, and that severely compresses the lumbar spine and angles the hips, leaving 20% of the entire breed affected by hip dysplasia. The craziest part? Von Stephanetz explicitly warned people back in the 1930s that selecting for appearance over function would destroy the dog. And sadly, his warning was entirely ignored. What's incredibly frustrating is that sometimes the science is absolutely there to fix the problem, but the institutions just refuse. In 1973, a geneticist actually fixed that fatal uric acid defect in Dalmatians we talked about earlier. He did it by crossing a Dalmatian with a pointer, which introduced a single healthy gene. By the 5th generation, the dogs looked exactly like purebred Dalmatians, but they were perfectly healthy. But get this,
In 1981, the American Kennel Club and the Dalmatian Club actively blocked the recognition of these healthy dogs. Why? Just to maintain the quote-unquote integrity of a closed gene pool. It took 30 years until 2011 for that block to finally be lifted. Just wild.
Section 3. Engineered for the farm. Okay, so if you thought the pet industry was concerning, wait until you hear about the agricultural models. This is where extreme genetic modification is executed purely for profit, and it's chillingly calculated. Consider this quote. That is not a complication. It is a design feature. That perfectly summarizes the reality of the Belgian blue cow. This is an animal selectively bred for a mutation in the myostatin gene. That gene basically tells the body to never, ever stop building muscle. The resulting cattle produce 40% more muscle than a natural animal. And because of this extreme artificial growth, the calves actually grow far too large in the womb to ever be born naturally. The exact cost of that so-called design feature is incredibly stark. 99% of Belgian blue births are handled via scheduled C-section. Natural birth is basically non-existent for these animals. These cows are intentionally bred to produce an animal that literally cannot physically enter the world the way animals have for millions of years. And the industry doesn't view this as a tragic medical complication. Nope, it's an accepted, priced-in, foundational business model for the Belgian beef industry. Section 4. Natural versus forced bottlenecks. We're going to shift our focus to wild animals for a minute to contrast natural genetic bottlenecks with ones that are caused or severely exacerbated by humans. Normally, a healthy immune system spots cancer cells easily because they look different and there attacks them. But contrast that with the Tasmanian devil. Due to really aggressive hunting by European settlers in the 19th century, their population completely crashed. The population that eventually grew back from that bottleneck was so genetically compressed that the animals are essentially identical at a cellular level. So when a contagious facial cancer appeared in 1996, the animals' immune systems literally couldn't tell the difference between the tumor cells and their own tissue. The cancer spreads uncontrollably just through biting, wiping out an estimated 60 to 80% of the wild population. It's a deeply tragic, delayed consequence of human hunting decades prior. Now let's look at the cheetah, an animal facing what we can call a double bottleneck. First, there was an ancient bottleneck about 12,000 years ago that severely reduced their initial genetic diversity. It's actually so extreme that totally unrelated wild cheetahs will accept skin grafts from each other like they're identical twins. Plus, 70% of their sperm is structurally abnormal. Now, the crucial point here is that humans obviously didn't cause that ancient bottleneck. However, our 20th century habitat compression forced this already incredibly fragile species into much smaller, isolated territories, effectively pushing them to the absolute edge of biological viability. But human intervention isn't always purely destructive. Sometimes we can actually fix the very bottlenecks we create. Take the Florida panther. By 1992, the remaining population was down to just 20 to 30 panthers, and they all had identical genetic defects. We're talking kinked tails, heart defects, and functional extinction was predicted within just a few decades. But in 1995, U.S. Fish and Wildlife stepped in. They released 8 female pumas from Texas into Florida to introduce outside genetic variation. And guess what? It worked. The hybrid offspring absolutely thrived, the universal defects completely disappeared, and the population was saved from total collapse. So we've clearly seen that breaking isolation can save a species. But that leads us directly to a really critical question. What happens when humans deliberately force the bottleneck? What happens when isolation isn't just an accident of geography, but the entire goal of the breeding program?
Section 5. The white tiger tragedy.
This brings us to what is perhaps the most disturbing example in this entire explainer of human greed driving deliberate genetic ruin. Back in 1951, Mohan, the last wild white tiger, was captured in India. Now, a white coat only appears when an animal carries 2 copies of a specific recessive gene. To reliably produce white offspring, both parents must carry it. To guarantee this happened, Mohan's handlers did the unthinkable. They bred him to his own daughter. The origins of the entire captive white tiger population rely on this absolute requirement of locking in a recessive trait, a requirement that mandated extreme, continuous incest generation after generation. The grim reality of that continuous inbreeding is stark. 1 in 30. Today, only 1 in 30 white tiger cubs is born without significant physical deformities. Let that sink in for a second. Every single white tiger born in captivity since Mohan is a product of that exact same closed system. The relentless pursuit of a rare, cool-looking coat color resulted in an astonishing rate of biological failure. And this brings us to the incredibly tragic story of Kenny the tiger. Born in 1998 to parents who were full siblings, Kenny had a severely compressed face, extremely misaligned teeth, and actually couldn't even fully close his mouth. He was originally going to be euthanized at birth, but he was kept alive purely because the breeder's son thought he was cute. Kenny eventually died of cancer at just 10 years old. That is far, far short of a wild tiger's natural 25-year lifespan. Kenny perfectly and heartbreakingly illustrates the intensely cruel consequences of breeding living creatures strictly for human entertainment.
Which leaves us with a final and honestly pretty provocative question. Are we preserving these animals, or are we only preserving our own destructive designs? From the racetrack to the laboratory, from the agricultural farm all the way to the pets sitting right there in our living rooms, the evidence is pretty clear. When we select for extreme appearance or extreme utility over health and function, the animals pay the ultimate price. I really hope this explainer challenges you to rethink what it actually means when we call an animal purebred or perfect. Keep digging and never stop asking the hard questions.